All right. Good afternoon, everyone. So can everyone hear me? Okay. So we're going to continue and wrap up this section where we're discussing antibiotics and kind of going along with the modes of action that we were talking about before. The next major mode of action for antibiotics that we're going to cover is interference with metabolism. Now, the way that it's written here, it's kind of a misconception here. And when I say interference with metabolism, I'm not talking about interfering with ATP production, but I'm talking really more about interfering with a metabolic pathway where inside of a cell, something needs to be produced. And for that something to be produced, it takes multiple steps for that to happen. So let's say that, well, I'm going to have to redraw this, all right, because this didn't quite work out the way that I wanted. So let's say that inside of the cell, I have some sort of product that I need to make. So right now, I'm just going to call this product D. Okay, so this is going to be my final product here. And I need to make that, but I can't just make it out of thin air. I need to make it in multiple steps. And in order to begin the process of making it, I'm going to start off with some substrate called A. So I'm going to there, but I can't convert A directly into D. The regular pathway that I have to follow will go something like this, where I have to convert A into some substrate B. And then after I've made the substrate B, I have to make C. And then finally, in a third step, I can take C and convert that into D. So I have multiple steps here. So we call this a metabolic pathway where we're using these multiple steps to do this. Now, each step in the production of this final product is regulated by its own enzyme. Okay, now, without going through all the different characteristics of enzymes, we know that enzymes are proteins and that all enzymes have a very unique structure. Did I talk about this? Did I go through this last time? Oh, okay. Well, great. Okay, so I thought that saves me a lot of time. I'm sorry about that. Okay. Okay. So I talked about sulfa drugs. All right, so we'll just move right along then. Give me a second here. Okay. So I think I had stopped with talking about I didn't talk about the nucleic acid synthesis inhibitor, so we'll pick up with that. Okay. All right, so sorry about that. So we're going to talk about antibiotics that inhibit nucleic acid synthesis. All right, so what we're really looking at here are antibiotics that either inhibit DNA replication or transcription. So remember that in DNA replication, we are making a copy of DNA, which all cells have to do before they divide. And in transcription, we're talking about the process of converting DNA into RNA. So the problem with a lot of these types of antibiotics that either inhibit DNA replication or transcription is that especially the very first ones that were produced, they were pretty toxic. The reason is that a lot of the enzymes that bacteria use to do a lot of these things, we have them too. But with that being said, there are some exceptions. And the exceptions that I want to talk about are a class of drugs referred to as the quinolones and something called rifampin. All right. So these antibiotics, they target the enzymes in bacterial cells and not our cells. So we'll start off here by talking about the quinolones. Quinolones are also a type of drug that's in that category. Might also be referred to as ciprofloxacin or Cipro, you may have heard of. And what this drug does, it blocks the DNA gyrase enzyme. Now, I know it's been a while, but does anybody remember what does DNA gyrase do? What is the normal function of DNA gyrase? Right. So remember that double-stranded DNA is in a double helix, and you have this helix structure. And one of the first steps in making a copy of DNA is you have to unwind the DNA and then separate the strands to make copies of it. So DNA gyrase unwinds the DNA. So if we block this enzyme, then the DNA stays permanently wound. Thus, you can't unwind it. You can't make copies of the DNA, and so it blocks DNA replication. Okay, so that's the main mode of action for this. All right, now it can be used against several different things, so it's kind of hard to say like it's only used against this and it's only used against this. But it does have a side effect that you do have to be worried about. So one of these side effects is that it can inhibit cartilage development. So for that reason, it's not a good drug to give newborns because, as you may well remember, newborn babies have a lot of cartilage in their skeleton, and that's important for bone development. And so it could inhibit that. All right. Now, the next antibiotic is rifampin. Actually, this antibiotic I've seen with different names. Most of the time, I've seen it called rifampin. It's also called rifamycin. A couple other names here, but we're going to call it rifampin here. This enzyme blocks transcription. It inhibits transcription in bacteria, and it specifically inhibits the process in bacteria. Okay, because what this enzyme does, it inhibits the RNA polymerase enzyme in bacteria. Now, we also have RNA polymerase to convert DNA into RNA, but this particular enzyme inhibits the bacterial RNA polymerase and not ours. Okay, so that's basically what it does. It's fairly broad spectrum, but to kind of narrow down what this antibiotic targets, there are some specific uses for it. One of the major specific uses for rifampin is it's one of the antibiotics that's used as a combination treatment to treat tuberculosis. Okay. Actually, let me move on here. If a person has tuberculosis, they are put on what we call the first line of defense drugs against TB. And there's about 3 or 4 drugs that are typically given. A person with TB would always get isoniazid and ethambutol. Now, does anybody remember? I know it was just last time that we met, but does anybody remember what is the target of isoniazid and ethambutol? Why would you want to give that? Okay, so yeah, isoniazid and ethambutol, their targets are the mycolic acids. So the idea here is that during the treatment here, if we have to get rid of the mycolic acids, because if we don't, none of the other antibiotics that we try to give will penetrate the cell. So you give these two at the same time to prevent the mycolic acids. Then you also give rifampin, or as you can see here, it's called rifamycin, same thing. And rifampicin blocks transcription. And a lot of times you give another drug called streptomycin, which inhibits protein synthesis. The idea here is that combination therapy is very common where you're giving multiple drugs where each drug targets something different. And by doing that, it helps to ensure that you effectively kill the organisms. So it's used, that's one of its major uses. Another use for rifampin, rifamycin, whatever you want to call it, is to treat meningitis caused by Neisseria meningitidis. Okay, so Neisseria meningitidis is a common cause of meningitis in children. And not only can it be used to treat that type of meningitis, but it can also be used to prevent it. So let's say that one person, let's say a child in a family has meningitis caused by Neisseria meningitidis, but the other people in the family are not infected, then doctors will would also prescribe rifampin to the uninfected family members to prevent them from getting sick. Okay, so it's used for those two specific cases. There is a odd side effect to taking rifampin, and it's something that's called red man syndrome. Um, Red man syndrome seems to occur if rifampin is given too quickly, like in an injection within 10 seconds, you know. Um, and they're not exactly sure why it might be happening, but it seems like if you give it too quickly, it causes a massive inflammatory reaction where a person's mast cells and basophils, they start releasing all this histamine that causes skin to get red. Sometimes even the person's urine gets red. And to avoid this from happening, if a person needs rifampin, it has to be given slowly over a longer period of time. So rather than 10 seconds or whatever, it's got to be given over like an hour or so, okay, to prevent this inflammatory reaction from happening. Okay, so that takes care of all of the antibacterial antibiotics. Um, what we need to do now is talk about some antiviral medications, okay? So a lot of these antiviral medications, they inhibit one or more steps of the replication process. You know, so when we talked about viral replication in the virus section, we talked about how there's attachment and then penetration, and then there's, you know, uncoding and then synthesis of DNA and RNA and protein, and then release of the virus. So a lot of these antibiotics, they're not antibiotics, the wrong term, antiviral agents, they block one or more of those different steps, okay? And to get started here, I want to talk about a class of antiviral agents that are known as base analogs. Sometimes they can also be called, I'll write this up here, a nucleoside analog. So big terms here, but essentially what a base analog or a nucleoside analog is doing is that incorporates a fake base into the genetic material. So of course they a fake base into the genome. So for example, okay, real quick, let's say that a virus's genome has got C, G, A, T, T, G, okay? And I want to inhibit the replication of this virus. So what I can do is I can give an antiviral agent that takes this, for example, this is just an example, takes this T and replaces it with a fake T. So it's a thymine, but it's not a real thymine. It's a fake thymine. It's like a synthetic fake thymine. And so when the virus moves to kind of make copies of this, it'll hit this fake thymine and stop. It won't go any further than that. And so you inhibit the replication of this. So essentially what these viral agents are doing is they're mutating the genetic material of the virus, preventing its replication. All right. So that's what's a base analog or a nucleoside analog essentially does. Now, one very common example of this is something that's really common, acyclovir. Acyclovir can be used to treat a lot of different types of herpes infections. So oral herpes, genital herpes, but not only that, chickenpox. Why chickenpox, you might ask? Because, well, the virus that causes chickenpox is in the same family. It's in the herpes viridae family, which is a big family. So any type of virus that's in that family, it can be potentially treated with acyclovir. Another antiviral agent that's also classified as a base analog is AZT. AZT is also a very common drug given to people that have HIV. And it's a base analog, but it's also classified as a reverse transcriptase inhibitor. Now, remember that HIV is a virus that we call a retrovirus. And what retroviruses do is they take RNA, they take single-stranded RNA, and they convert it into double-stranded DNA. So they're reversing the process of transcription. And to do that, they need this enzyme called reverse transcriptase. And this is something that the virus, it's part of the virus that brings it with it. Well, the way that AZT inhibits this enzyme, it doesn't attack the enzyme directly. But what it does, it mutates the RNA. It puts in a fake nucleotide here, a fake base, so that when the reverse transcriptase enzyme is trying to convert this RNA into DNA, it reads that fake base and it stops. And that prevents it from converting the RNA into DNA. So that's how it shuts that down. All right, now here's an antiviral agent that is actually not used anymore, but I do want to talk about it. It's a drug called amantadine, and it used to be used to treat influenza, the virus that causes the flu. The reason it's not used anymore is because it was used so much that most viruses have developed resistance against it, okay? But what it does, it blocks the uncoding step. So remember that when a virus enters a cell, after it enters the cell, it has to separate the capsid from the nucleic acid, and so that the host cell can have access to the nucleic acids and make copies of it and make proteins from it. Well, this antiviral agent prevents that. So the influenza virus's genetic material stays permanently inside of the capsid, and eventually it just disintegrates and is no longer viable. Okay, so it blocks the uncoding step. All right. Now, there's another class of antiviral agents that are also typically given to people that are infected with HIV, and they're called protease inhibitors. Okay, so what a protease inhibitor typically does, let me erase this real quick. So again, it's given to HIV. So remember that HIV is a retrovirus. So when retroviruses replicate, they convert single-stranded RNA into double-stranded DNA. Okay, and the reason that they're doing that is now, once they've got that DNA, they're going to take that DNA and they're going to insert that DNA into your DNA. Okay, so in black, this is the host DNA. And in here, this is the viral DNA. And the reason that these retroviruses do that is to basically camouflage themselves. They're trying to make themselves look like they're part of you. Okay, but the whole time that this virus's DNA is just stuck inside your DNA, it can continue to make new viruses. It'll make more copies of its genetic material, and it'll also make more proteins. Now, it's the protein production part that's a little strange with retroviruses, because with a lot of other viruses, when they make their proteins, they make all of their proteins individually, separated from each other. But retroviruses don't do that. With retroviruses, when their proteins are initially made, they're all connected to each other. So for example, if we look at something like HIV, it would have, the protein for, like, all the proteins needed for the capsid proteins, and attached to the capsid proteins, you would have the spike proteins connected to that. And then attached to that, you would have, for example, the reverse transcriptase enzyme attached to that. So it produces what's called a polyprotein. Then later on, after these polyproteins are produced, an enzyme called a protease comes along and cuts these proteins apart from each other. So it's like we're coming in here and saying, okay, clip, clip. And once these proteins are separated from each other, then you can use them to build a mature virus. Well, a protease inhibitor inhibits that enzyme. And when that happens, these proteins remain permanently attached to each other. When these proteins are permanently attached to each other like this, the virus cannot use them to make and replicate a mature virus. So we inhibit the replication process that way. So, and I know there's a lot of different antiviral medications that are new and upcoming that are being given to people with HIV, but typically they're given AZT on the one hand to prevent, or something very similar to that, that prevents reverse transcriptase enzyme, and then protease inhibitors to inhibit this process so that you're trying to, again, hit multiple targets to prevent replication of the virus. So that's kind of how it goes. All right. Go back here. Another antiviral medication that can be given is interferon. Now, we've talked about interferon before because we know that interferon is a chemical. It's basically a chemical messenger that's produced by cells in your body that are infected with the virus. Now, the chemical doesn't save the cell that's making it, but it might be able to help the surrounding cells, the neighboring cells, so that if they're ever infected with that same virus, they can protect themselves. And for certain viral infections, interferon can be given in IV fluids and injection. For example, hepatitis B, you can treat that with interferon, or hepatitis C infections, and there's other viral infections that can be, possibly treated with interferon. Okay, now I mentioned that this drug called amantadine that used to be used to treat influenza, and it's not used anymore because most influenza strains have developed resistance against them, unfortunately. But there is another anti-influenza drug that has been produced that's used. It's called Tamiflu. Now, Tamiflu does work, although if you've been diagnosed with the flu, you have to take this within about 24 hours from the onset of the symptoms, because after 24 hours, it doesn't really work. Okay. For whatever reason, it just doesn't work. It's probably due to the fact that the virus has spread so much that it doesn't work. It doesn't really, it's kind of useless or pointless. So Tamiflu is described as an antiviral agent that is a neuraminidase inhibitor. Now, I know that doesn't mean anything to you, but let me explain what it's getting at. So down here at the bottom, you see a very basic diagram of the influenza virus, and it has all these little spike proteins on its surface. Well, there are two spike proteins that are important for this virus, and that's what I want to talk about. So one of these spike proteins is called the hemagglutinin, or H spike, and the other one is called the neuraminidase, or N spike. So we have an H and N spike. The H spike is used by the virus for attachment. So when this virus attaches to the cells in your body, it uses that spike to specifically attach to it. And then it gets into the cell, uses the cell to make more of itself. Now, the next, after numerous new influenza viruses are produced, then these viruses have got to be released from the host cell. And since this influenza virus it's an envelope virus, it's going to be released by this budding process. I mean, that's basically how it's going to get its envelope. So what happens is that as the virus is budding off of the host cell, initially it remains attached to the host cell. There's like this protein that kind of chains the virus to the host cell. To release the virus uses the N spike, the neuraminidase. And if you look at the word here, it ends in ACE, which is an enzyme. And what this enzyme is used by the virus to do is to basically it basically cuts that protein that's chaining the virus to the cell, kind of cuts that away, releasing the virus from the host cell, and now the virus can go on and infect other cells. So what Tamiflu does as a neuraminidase inhibitor, it basically inhibits that enzyme. When that happens, the virus remains attached to the host cell. And eventually, the more and more it kind of stays there and doesn't get released from it, it disintegrates and it's not viable anymore. All right. And that thereby prevents the spread of the virus to all the other cells in your body. Okay, so that is how Tamiflu works. Okay, so so that's all the antiviral medications. Now, I want to talk about some medications that can affect and be used against other things. So how about some antiprotozoan medications? And we want to talk about chloroquine, or any drug that has quinine in it, or a derivative of quinine. Well, chloroquine or quinine is generally used to treat malaria. How does it work? We're not really sure. It's thought that quinine blocks metabolism somehow, but we're not really sure. But we just know it works. And quinine can be used to not only treat malaria, but if, let's say, you're planning on traveling to a country that has a high rate of malaria, a few months before you go, you might want to tell your doctor that you're going to this country, and is there anything you can do? What your doctor will probably do is put you on chloroquine or quinine, so that when you do travel that country, and if you are bitten by a mosquito that's carrying Plasmodium, you'll already have the medication inside of you so that it won't replicate, it won't spread. Okay, so that's one of the general uses for it. Metronidazole. One of the primary uses for this is to treat Trichomonas vaginalis infections. Remember, Trichomonas vaginalis is a protozoan that is unique to humans, and it causes a sexually transmitted disease called trichomoniasis. And this is the antiprotozoan medication that is used for that. All right, well, how do we fight off worm infections? And there's a lot of different medications that can be used for worm infections. We're going to just kind of focus on two here. So anti-Helminth medications. This first one, niclosamide, is typically used against tapeworms. And it's thought that niclosamide inhibits or shuts down the electron transport chain in the worm. So the idea there is, if there's no electron transport chain, there's no ATP. If there's no ATP, there's no energy, and the worm dies. All right, praziquantel. This particular anti-worm medication can actually be used for a lot of different worms. It can be used for tapeworms and pinworms and other types of roundworms as well. And what this antibiotic, I keep saying antibiotic, what this anti-Helminth drug does is that it causes the muscles within the worm to permanently stay in a contracted state. And when they're permanently contracted, the worm can't move and they're effectively paralyzed. How does it do that? Well, without going into all the specifics that you went through in A&P1 about muscle physiology, you hopefully remember that calcium is needed to get muscles to contract. Without calcium, muscles can't contract. So worms also have muscles that they use to move around. And what this drug does, it causes a massive amount of calcium to enter the cell. And when there's a massive amount of this calcium, it causes sustained muscle contractions, effectively paralyzing the worm. All right. Okay, how about some antifungal agents? So how do we prevent the growth of molds and yeasts? Well, there's a problem with this, because molds and yeasts are eukaryotes, and so are we. And a lot of the initial antifungal agents that were discovered were very toxic to us because they attacked a lot of the same things that we have. But there are a couple of notable exceptions to this. One of the exceptions takes advantage of the fact that fungal organisms have ergosterol in their cell membranes. Whereas you and me, in our cell membranes, we don't have ergosterol, we have cholesterol. So some of the anti-fungal agents will attack ergosterol in the cell walls of fungi and not the cholesterol in us. Now, there is some cross-reactivity. There can be some toxicity associated with it because cholesterol and ergosterol look very similar. And, depending on your use, there might be some toxicities, and some people are more sensitive to it than others. But for the most part, these antifungal agents that I'll talk about are pretty good at targeting the ergosterol in fungal organisms and not the cholesterol in us. So here's kind of a list of some of the antifungal medications here. So the first three that are within this red square, amphotericin B, miconazole, and terbinafine, the targets of those drugs are the ergosterol. Okay, they might be used for different applications, but their target is the ergosterol in the cell membrane of these fungal agents. So let's start off here with amphotericin B. Now, amphotericin B is typically given to treat systemic fungal infections. So if a person is having a fungal infection in their respiratory tract or in their nervous system, or in their cardiovascular system, or any sort of body system, to really penetrate those tissues and those body systems really well, amphotericin B is given for that. Okay, and what it does, it actually finds the ergosterol in those organisms and destroys it, and leaves a hole in the cell wall where there used to be ergosterol. And when that happens, now the fungus will absorb fluids from the outside and swell up and pop by lysis. The other two, miconazole and terbinafine, they work similarly. Typically, miconazole and terbinafine are typically given as like topical ointments to treat a lot of topical infections, but they work by inhibiting the synthesis of ergosterol. Now, the other two antifungal agents that you see down there, they work completely differently. They are antifungal agents, but they work in a different way. Griseofulvin inhibits mitosis in fungal organisms. You might remember that in mitosis, you have to produce a mitotic spindle, which is made of microtubules that separate the chromosomes apart from each other. And griseofulvin prevents the mitotic spindle from happening. So no mitotic spindle, no separation of the chromosomes, no division. Okay. And then the echinocandins. The echinocandins inhibit the fungal cell wall production. Now, does anybody remember, what is the cell wall of a fungus made of? Right, it's made up of this really, really tough carbohydrate called chitin. All right, chitin. And one of the sugars that's in chitin is glucan. Without glucan, you can't make chitin. So the echinocandins prevent the glucan synthesis. So basically, you're inhibiting the cell wall that way. Okay, so I'm pretty much done talking about all the antibiotics, antiviral agents, all this kind of stuff. So I want to talk about a couple other topics here. One, if we're trying to test out whether a certain antibiotic is effective in inhibiting the growth of certain organisms, how can we do that? Well, for bacteria, what you would use is something called a disk diffusion method. And y'all have done this in the lab, where you would make a spread plate of whatever bacteria you're interested in, and then on top of the spread plate, you would place a paper disc that's impregnated with a certain antibiotic. Then you would incubate it, and if that antibiotic is effective in inhibiting the growth of the bacteria, what you would see is a zone of inhibition. Okay, it's basically an area around a disc like this where there's no bacterial growth. And the general idea is that the larger the zone of inhibition, the more effective that antibiotic is. All right, so that's one of the ways that we test for that. Now, before we're done today, I do want to talk a little bit about, well, why do microorganisms become resistant against certain antibiotics and anti-micro medications? Because we're seeing more and more organisms becoming resistant against a lot of the medications that we use to treat them. And the question is why? Well, it's very simple. The more and more that microbes are exposed to these things, eventually they evolve a method to, evolve a way to live in their presence. It's really all about evolution. It's survival of the fittest, where they try to find ways to survive in the presence of these antibiotics. And if they can find a way to do that, then they have a survival advantage. They're more likely to persist and cause infection. Now, one thing that's causing a lot of this is just overexposure to antibiotics. So, when a person is infected with a bacteria, for example, you go to the doctor and the doctor gives you an antibiotic. It's the normal thing that they do, but it's happening so much that the more and more that these bacteria are exposed to it, they eventually, and we're seeing it right now, they develop resistances against it. The other thing is that you don't necessarily have to be taking an antibiotic prescribed to by your doctor to be exposed by it. Studies have found that a lot of the meat that we use, that we eat for, like chicken and beef and other types of meats have got antibiotics in them. These animals are fed antibiotics to prevent infection in them. So when we eat them, we're basically taking in those antibiotics indirectly. Another way that bacteria can become resistant is, let's say that your doctor says, okay, you have to take 10 days of an antibiotic twice a day to really cure this infection. And you start taking it and you're pretty good at it. But then after like 5 days, you're like, you know what? I feel better. I don't think I need this antibiotic. I need to stop. And the problem with that is if you stop, you're not really killing all of the bacteria that were inside of you. And so the ones that are left, they're going to be like, it's not like they really think about it, but they're like, we need to find a way to survive in the presence of this antibiotic so that if we're ever exposed to it again, we can still infect this person. So they're going to mutate. They're going to find a way and be resistant against that antibiotic so that if you have to take that antibiotic again, it ain't going to work. Okay. So these are some of the things that we are doing or that we're being exposed to that do this. Another example, antibiotic soap. We may have talked about this before. A lot of people choose to buy that has antibiotics in them. You do not need antibiotic soap because you're just increasing the exposure. You're just increasing how much these bacteria are exposed to it. So it might work now, but the more and more that these bacteria are exposed to it, eventually the antibiotic in the soap doesn't work and they develop resistance even against other antibiotics. Okay. So we are creating superbugs, whether we know it or not. And then once they've become resistant, let's kind of review. And I've been doing this. We've been talking about this as we've been going through this. But when they become resistant, what have they done to become resistant? What have they made that is making them resistant? So one thing is they can alter or prevent the antibiotic from getting in. One example of this is just take a look at the difference between gram-positive bacteria and gram-negative bacteria. And we've talked about this before. In general, which types of bacteria are harder to kill, gram-positive or gram-negative? Gram-negatives, because they have that outer membrane that serves as a protective barrier. Another example, mycobacteria, or any other acid-fast bacteria, the mycolic acids prevent them from getting in. Okay, so they can prevent the antibiotic from getting in. Another example of this, of how a bacterial cell can prevent the entrance of a antibiotic, there are some tricky bacteria that if they're exposed to an antibiotic enough, they evolve the production of a pump system. It's like a little pump. And what they use the pump for is that if they're exposed to that antibiotic, the antibiotic gets in, but then the pump pumps it straight back out. So the bacteria doesn't die. All right. Another thing that bacteria can develop to become resistant is a way to inactivate or destroy the antibiotic. We've talked about an enzyme that some bacteria produce that can destroy certain types of antibiotics. Does anybody remember the name of that enzyme? Well, it's not the blah gene, but the enzyme, they would have to have that gene to do this, but you're close. So they would produce that enzyme called beta lactamase. Okay. And I haven't talked about a transcriptase. I'm not sure what that is. But a beta lactamase destroys beta lactam drugs like penicillin and amoxicillin, things like that. Some bacteria will change the target of the antibiotic. So, for example, if I have an antibiotic that targets the ribosome in bacteria, and these bacteria are exposed to this antibiotic over and over again, then what the bacteria can do is over time, they can change, they can mutate and change the structure of the ribosome so that the antibiotic doesn't bind to it anymore. All right. And then finally, another thing that bacteria can do is replace one metabolic pathway with another one to be able to still make some sort of final product. So this kind of relates to when we talk about the sulfa drugs preventing folic acid synthesis, where we had that multiple step process where we go from some substrate A to some substrate B to C, and then finally to folic acid, right? And then we talked about how the sulfa drugs, they block the first enzyme in the pathway. And you also typically give trimethoprim, which blocks the third enzyme in the pathway. But with overuse of these antibiotics, what these bacteria can eventually do is find another way to get from here to here. They might evolve the production of different enzymes and different metabolic pathways to finally be able to make folic acid. All right. So yeah, bacteria and these microbes, they don't have brains, but they're very smart. They evolve different ways to survive. Okay. All right. Well, I am done with this section. We're done with antibiotics and all these different medications here. So I'm not going to start on anything new, but on Monday when we come back, we will start with respiratory system diseases. Does anybody have any final questions before we call it a day? So we'll end early today, but before we do, does anybody have any questions about anything? Yes, Rose, go ahead.
I just wanted to confirm for the review, some of the diseases like cardiovascular and gastrointestinal, those will be on the final and not test 4?
That is correct. So test
review.
4, test 4 will cover antibiotics, respiratory diseases, skin diseases, and nervous diseases. And then after test 4, we're not quite done yet. We still have to lecture on cardiovascular, GI diseases, and genitourinary diseases. But questions on those last 3 sections will appear on the final exam. All right. So the final exam is going to have questions from those last 3 sections and questions from all the other stuff that we talked about this semester.
Okay. Thank you.
Okay. Yeah. Any other questions? 56. Let me see. 56 here. This one? Okay. Anything else? What did I say about the sulfa drugs? Was it something specific? I mean, kind of going back here, and I'm going to have to switch slides here. If we go back to what I stopped with last on Monday. Well, the sulfa drugs, they inhibit folic acid synthesis in bacteria. So this is folic acid here, and to get to folic acid, there's a 3-step process where you have to use enzyme 1 that regulates the first step, enzyme 2 that regulates the second, and enzyme 3 that regulates the third. And sulfa drugs, they they basically inhibit the first enzyme of the pathway. But you always give trimethoprim at the same time, which blocks the third enzyme of the pathway. So, but I did, yeah. No, no. Okay. All right. So I will see y'all Monday, and hope you have a good weekend. And as always, if you have any questions, feel free to email me. All right. Thank you very much.